I'd like to introduce Kerry. She's a professor in deep sea ecology from the University of Plymouth in the UK. And she has a a bachelor's degree in marine biology and a PhD in deep sea ecology. And she has over 20 years experience researching benthic fauna distribution patterns in the Atlantic deep sea. Her research is focused on mapping benthic biological communities to support sustainable management of deep ocean and integrating habitat maps into marine spatial planning processes, including marine protected area network design. And the way we linked up is that she is actually one of the co-directors of the One Ocean Hub program, which Bronwyn was talking about way back when. I think it was January or February. And the aim of this project is to basically address the global challenges in sustainable ocean management. And she's also a partner in the EU-funded Mission Atlantic project. So Kerry, please share your screen and go ahead. Thank you.
Well, thank you very much, Thibaut, for that nice introduction. And thank you so much for inviting me to speak today. I'm very grateful for you for your time. I'm going to talk to you today about some work that we're doing on benthic marine habitat mapping in the Southeast Atlantic, and particularly some work we're doing around vulnerable marine ecosystems. And this work is really focused around supporting spatial management of the oceans, and particularly the deep oceans. So mapping really lies at the heart of spatial management, and it might be maps of things like human uses of the marine environment. So it might be maps of fishing effort, or it might be maps of spatial maps of resources, so things like oil and gas license blocks. But when you're thinking about the ecosystem approach to marine environmental management, one of the most important layers of information that you need to include is information on the biology and the habitats that you have. So we need to think about making habitat maps. And those, put really simply, are give us an understanding of what we have in terms of our biological resource and where it is. So it's really quite basic, but it's actually fundamental to spatial management. And this is particularly the case when thinking about things like vulnerable marine ecosystems, where it's really important that we understand where they are in space in relation to things like fishing effort and bottom trawling activities, if we're trying to be sustainable in the way that we use our ocean. And habitat is a funny word because it means different things to different people, depending on who you're speaking to. And it also operates at different scales. So you can consider habitat maps at different spatial scales. So you might think about things at the very whole ecosystem level, or you might think about it on the scale of smaller communities of organisms like vulnerable marine ecosystems, or you may even think about it on the scale of individual species. Where are they, and how are they distributed? But in any mapping approach that you take, that mapping must be underpinned by a biological understanding of where things are. And so it needs to have, feeding into that process, biological information. And this is possibly one of the biggest challenges in the South Atlantic, is the lack of biological data. So the Ocean Biogeographic Information System, which is maintained as a sort of UN level database, which contains information on where people have sampled and biological data. If you extract information from OBIS and you only consider deep sea species, so that's things below about 200 meters, this is what you get. And what you can see from this is that there is a very big hole in our knowledge from the Central and South Atlantic, as well as other parts of the world, but let's focus on the Atlantic, in comparison to the North Atlantic, which is relatively data rich. And this extract actually includes a lot of pelagic data. And in fact, a lot of what you can see for the South Atlantic is in fact pelagic information around fish species and also physical measurements. When it comes to benthic data, there's even less than there would appear to be here. So we have a problem in that we don't have a lot of data, and yet we are using our marine environment, and we would like to seek to be responsible in the way in which we use it. And so the question then becomes, how can we produce meaningful maps with limited biological data? And this is true for lots of areas of the deep sea around the world, but it's a particularly acute problem in the South Atlantic. So there are various ways in which we can produce habitat maps using limited biological data, or even in this case, no biological data. So one of the most common approaches is to map environmental variation as a proxy for biological variation. And the simplest way to think about this is that if we think in terms of biogeography, which is this layer of information here, we know, for example, that the animals that you find living in the benthos in the Arctic or the Antarctic are completely different from the animals you will find living in the Atlantic, for example. And so at a very, very coarse scale, at a global scale, we can use information on biogeography to delineate different areas of the ocean that contain different animals. And so this is a very simple biogeography, but you can get the idea that here is a map which represents variation in the biology. It's a very coarse scale variation. But we can use this premise with other environmental variables. So we know the presence of organisms is dependent on temperature. So things only have specific thermal regimes that they can live in. We know substrate type is important to marine benthic organisms. Things that live on rock are not the same as things that live in and on the sediments. And we know that organism composition, community composition, varies with water mass structure. And that also relates to variation in faunal communities with depth. So things that live in the bathyal are not the same as things that live in the abyssal. And we actually have a continuous turnover of fauna with depth. So what we can do, if we feel we understand what are those key environmental variables that shape the biological community patterns that we see, we can use these environmental variables as proxies for the biology, and we can map environmental niches is what we're really mapping, ecological niches, and those are used then to represent biological variation. Now, this is not a new approach. This approach has been taken by a number of nations. So here you can see a map from Australia. This is Australia's national marine habitat map. And that's freely available online. Here is an example from closer to home. This is the South African National Marine Habitat Map. And again, the concept of each of these habitat maps is that the different coloured classes on the seafloor represent different biological communities. That's the whole premise for these habitat maps. So this is the South African one. And over in Brazil, they've taken a similar approach. They have sectored up their EEZ into different regions and produced habitat maps for those different regions using this sort of approach. And this is really important. So individual nations have been taking this sort of approach and producing their own habitat maps, and long may that continue. Individual nations know best their environment. They also understand most about what they need from those habitat maps in terms of how it's going to inform their governing processes. And they also are most likely to have the highest resolution environmental data available to them for that region. And so those national habitat maps are very detailed and very important for those nations. However, the problem comes when you are starting to think about managing larger regions. So areas like the Benguela upwelling system, or the large marine ecosystem covered by the Benguela Current Commission, for example. If you try to put together habitat maps made by different nations to have an overview of what's happening in a region, the fact that those maps have been made using different methods different data inputs in many cases. They are often built at different scales or different resolutions of data. And the result is that you have different maps. And it makes it very difficult to put these things together to say anything useful on that regional level. And in my work, I'm concerned particularly with areas beyond national jurisdiction, and trying to manage those large areas and think about those transboundary issues with how they connect to nations' EEZs means that it is a problem if you're trying to put multiple different habitat maps together. And so some work that we've been involved in is then trying to generate a habitat map for the whole of the South Atlantic, predominantly to look at management in A, B, and J, but actually it can serve as a base for other initiatives within the region if it proves to be useful. And so we're working on this with a number of South Atlantic facing nations and partners to try to produce something that is biologically meaningful for the region and potentially useful for nations, but certainly within A, B, and J. So we're taking two approaches to this because there are lots of ways of producing these maps, and no one way is more correct than the other. It's just different ways of modeling. So we're using an approach that is very data-driven, shall we say, and it's based on a couple of publications that have come out, and it's really based on a type of principal component analysis of environmental variables, and then modeling using the principal components to divide up your ecosystem into different ecological niches. And then a second approach, which is based more on a classification type analysis, which is based on work that I have done in the North Atlantic in the high seas area, but also my student, Kirsty McQuaid, who's a South African PhD student, has been working with me. She has taken this approach in the Pacific, in the Clarion-Clipperton fracture zone, working in A, B, and J there, and looking at deep sea mining related issues. So there's these two approaches, PCA type approach, which we're calling the data-driven approach, and a cluster analysis type approach, which is also data-driven, but it's a different type of analysis process. So we are just in the process of doing this work. So the map I'm showing you here is not the map. This is a draft output, hot off the press. We have not even been able to share this with the project partners yet, but I wanted to provide you with an illustration of the sort of output that you get. So this is not the output, but it is one of the scenarios that we've run so far in our tinkering with these methodologies. And so this is a habitat map it actually is for the whole of the Atlantic, but we're focusing on the South Atlantic here. And as I've said, we're working with partners in South Atlantic facing nations with the intention of producing something useful, but our project, the One Ocean Hub, is really considering areas beyond national jurisdiction. So just to have an initial look-see, and again, this is not the output, but I think it's interesting to have that very quick look. We did a very quick visual comparison here with the South African National Biodiversity Habitat Map, National Marine Habitat Map. So that's the one at the bottom here. Have I got my thing? I can't find it. There it is. So this is the national South African national map, and this is our model. The colors are different. I apologize for that. It was different output. But what I hope you can see is that in this particular model that we've run, there are some similarities. There are some clear differences as well, but certainly there are areas of congruence. So here you can see this sort of blue coloured area here in the national habitat classification system is sort of similar to this whiter coloured area up here, this pale area. There are some similarities on this coast as well, although the banding is not exactly following what it should. We can see the seamounts showing up here, and again we see them showing up here. And these two approaches were different approaches to mapping. But it's interesting to see there are some areas of agreement, but also some areas of difference. And so what we're hoping to do with this is, once we've produced our first draft models, we will share those with the partners who are working on this with us, and do this sort of comparison work where nations are happy for us to do that, and see how useful or not, and what where there are areas of agreement between these types of approaches. So that's one way of doing things, but I'd like to return to this question of how can we produce meaningful maps with limited biological data. So that approach really doesn't require any biological data. What it requires is an understanding of the environmental drivers of your biological variability. But there are times when you actually do need to know about the biology, and I'm talking now specifically about the example of vulnerable marine ecosystems. So for those of you who might not be aware, vulnerable marine ecosystems is a concept that emerged from discussions at the United Nations General Assembly. And VMEs are basically areas that are vulnerable to the impacts of bottom fishing activities. So that might be trawling or longlining or other methods. And states have a requirement on them to protect vulnerable marine ecosystems. Now, there are no strict definitions as to what is a vulnerable marine ecosystem. What there is a set of guidelines about what criteria an area has to meet, or a community of organisms has to meet, in order to be considered a vulnerable marine ecosystem. So these are those characteristics. They could be unique or rare. They may be functionally significant, so things that are necessary for the survival, function, spawning, reproduction of other organisms. They may be highly fragile, so easily susceptible to damage from anthropogenic activities. They may have life history traits that make recovery difficult, so they might be slow growing, have late maturation, or unpredictable recruitment, or be long-lived. Or they may be structurally complex in that they form a habitat for other species. And a vulnerable marine ecosystem does not have to meet all of these criteria. It only has to meet one. So different regional fisheries management organizations have actually have different interpretations of these guidelines. But let's consider there are some habitats, shall we say, that most RFMOs agree are vulnerable marine ecosystems, and that's things like cold water coral reef, sea pen aggregations, sea pen fields, deep sea sponge aggregations, xenophyphal aggregations, and things like coral gardens, which you can see just in the bottom right hand corner of the screen, and left hand corner actually. But also things like hydrothermal vents and cold seeps. So these are all kind of universally accepted as being vulnerable marine ecosystems. And so when you're thinking about fisheries management and mapping, it's really important that we understand the distribution of vulnerable marine ecosystems. Now that's actually quite challenging. But approach we've taken in the North Atlantic is to map the occurrence of what we call vulnerable marine ecosystem indicator taxa. So that's not the actual ecosystems, it's organisms that are common in those ecosystems. And it tends to be things like corals. So we've got here on the right hand side of the screen, black corals, cup corals, gorgonians, sea pens. So it tends to be corals and sponges. Chemosynthetic species are also included, excuse me. And so what we've tended to do in the North Atlantic is collect data on the occurrence of these taxa, and those data have largely come from fisheries bycatch actually. So all these sort of data points you see here, largely the ones running along the shelf, slope edge, you can see are actually coming from national fisheries monitoring programs, and these are bycatch, and they monitor that bycatch data. And it informs on VME indicator taxa, but we do also have data coming from the academic community and academic research in the region. And this approach is great. It's a really straightforward way of considering vulnerable marine ecosystems and mapping their distribution. But of course, it means you are largely limited to those areas where you have those monitoring programs, which tend to be the areas that are heavily fished, and therefore not necessarily the areas where those vulnerable marine ecosystems are still in good condition. And actually, it's really useful to have information from areas that are perhaps less fished. And so we've been looking to try and produce maps of the distribution of vulnerable marine ecosystems that are comprehensive, so they cover the whole area. And one way that we can do this is by using something called species distribution modeling, also called habitat suitability modeling. And this is based on the premise that if you know the environmental conditions under which you observe your vulnerable marine ecosystem, so in this case, cold water coral reef, you have observations of where you have seen cold water coral reef or you've encountered it, and you record the environmental conditions at that location, then you know something about the environmental preferences of that VME. Similarly, if you record where you haven't come across that vulnerable marine ecosystem, you know something about the conditions it does not occur under. And by formalizing the relationship between the presence and absence of your vulnerable marine ecosystem and the set of environmental conditions it does and doesn't occur under, you can formalize that in a statistical equation, and that enables you to then predict where you are highly likely to find your vulnerable marine ecosystem based on the environmental conditions of the area. So we have been doing this kind of work up in UK and Irish waters. So this is the UK and Irish continental shelf claim area. And we've been modeling the distribution of cold water coral reef and a type of deep sea sponge aggregation in this region. And what those models produce then are probability layers of the likelihood of you encountering your vulnerable marine ecosystem. So the red areas, you are highly likely to find it, and the blue areas, you're highly unlikely to find your vulnerable marine ecosystem. And you can turn these models into presence absence models, so you can turn them into yes-no maps of where your habitat is if you want to, using some statistical thresholding techniques. But actually, it's quite useful to leave it as probability because it gives you an understanding of risk. So we can do this in the North Atlantic because this area, known as the Rockall Trough and the Porcupine Sea Bight, are really data rich. We have been studying this deep sea area since deep sea science began, and we have a huge amount of data for this region. We also have very high resolution bathymetry for this data. So this is for this area, this is multi-beam bathymetry. And all of these points are presence or absence points for cold water coral reef in this region. So we've got a lot of data, and that enables us to build quite high resolution models for this region. But what we were asking ourselves is, actually, can we use areas of the deep sea that we know a lot about? Can we use these data-rich areas to actually tell us something about data-poor areas? So can we build models for the North Atlantic and transfer them to the South Atlantic and have them give us some useful products? Do they produce maps that reflect reality? Are they accurate maps? So that's one of the questions we've been addressing in the One Ocean Hub, is asking if we can actually do this. Does it work? Because if it does, it provides us with a means to produce maps in the South Atlantic that can be useful for spatial management in the absence of a huge amount of biological data. Now, that's not to say we don't want to collect that data, we absolutely do. But what we can do is use these models in the meantime, while we then set out to collect the biological data to validate them and build better models. So we've been transferring these models to the UK overseas territories in the South Atlantic. So Ascension Island, St Helena and Tristan da Cunha, which are A, B and C on the map here. And so we've been predicting onto these areas of multi-beam around these islands and seamounts in this region to produce models of the distribution of cold water coral reef as our first test area. But thanks to a collaboration with the Southeast Atlantic Fisheries Organisation, so CIAFO, and also the Nansen programme, we've been able to obtain multi-beam data for a number of the seamounts in ABNJ, just off the coasts then of just outside the Namibian and South African EEZs. You can see them all plotted here. And we've been able to make predictions in this area too about the presence of cold water coral reef on these different seamount regions. We have had a small amount of data that we've been able to validate these models with, coming from the UK overseas territories largely. And so our initial impression is that this model performs really quite well. And so this may be a useful way of mapping cold water coral reef, at least in the South Atlantic, which is really interesting. But unfortunately, this model is a high resolution model, so we are modeling onto multi-beam bathymetry. We can produce models based on the GEBCO bathymetry grids, which would mean we could cover the whole of the South Atlantic. But we know that those models based on the GEBCO data are less reliable. And so while they might provide us with an indication of where we should look for cold water coral reef, you certainly wouldn't use them as they are for management. You would want to probably go and look in those areas and then produce more detailed maps of where the cold water coral reef is within those high likelihood areas at a broad scale. So I want to come back to this issue of all models need data though. So, you know, a model is only ever as good as the information going into it. And the broad scale models I showed you at the beginning are based on global environmental data layers, which are themselves models. And we know that their accuracy in the South Atlantic is not that great. GEBCO is not great in the South Atlantic, particularly, and the global ocean models of oceanography, of current speeds, of temperature, salinity, these things are also still lacking data for the South Atlantic. And the other modeling approaches I showed you, so we can transfer models from the North to the South Atlantic, but we still need to validate them to know if they're any use, and that requires biological data on where things are and aren't. So we still do need the biological data. This is not a means to an end. You know, these models can help in the interim, but there really does need to be effort on collecting biological benthic data from this region. And I just want to finish by highlighting a hope I have for the next 10 years, I suppose. As you may know, we are now in the UN Decade of Ocean Science for Sustainable Development. We know there are huge data gaps in the Central and South Atlantic. And so I would hope that over the next 10 years, we can really focus on trying to fill those data gaps. And one of the ways in which we hope to do this is through a project called Challenger 150, which is a new 10-year deep sea science project, which is global in scope, but it has a Central and South Atlantic Scientific Research Committee. And through that committee, we are trying to coordinate scientific activities in the South Atlantic deep sea to, I guess, just be a bit more joined up about collection of data and really trying to, I guess, encourage people to collaborate on trying to get data from this region to help us better manage the deep water areas of the South and Central Atlantic. So I'll finish there. Thank you very much for listening, and I hope I haven't gone over time.
Thank you, Kerry.
I'm mostly interested, of course, the methodology that you do this. It's like satellites underwater, right? You have pictures from the ground, but you still need to trawl to collect all the animals and then assign them to the type of ecosystems that you have defined by your more or less remote sensing methodology. Is that correct?
Yes, absolutely. Yeah. So we still need the biological data to understand exactly in terms of the biology what those different ecological niches mean and represent, for sure. Those broad scale maps are useful, particularly for things like marine protected area network design, where you may be wanting to represent the different communities that you have in your region, and you might want to assess whether your MPA network is being representative of the different habitat types, in which case those different ecological niches can be, you can use those to assess whether you are representing each one of the niches. But yes, absolutely, to understand what they mean in terms of the biology, you still need the biological information. And I think that's, you know, that's what I mean by we still need the data. You know, we can make these maps and they have uses, but that doesn't get you away from the need for the biological data.
So are you going to trawl the whole South Atlantic?
No, absolutely not. No, but I think you do want to validate these models. So we do want to feel confident that the different classes we identify are truly biologically different and represent something. And then, yeah, of course, you can never sample everywhere. So then you have to rely on the model, but you should still validate your model and check, you know, what it means.
Thank you.
Gary, this
Hello.
is Dion. This is Dion from Namibia, Swakopmund.
Hi, Dion.
Can you hear me?
Yes.
Hi. I assume that what you just showed, it's just mostly for the deep sea environment and not very close to, you know, to shore. When you're talking about modeling, you know, what you're doing in the northern part, and try to do that in the southern part.
Yeah, the work we're doing is largely focused below 200 meters.
Okay.
Because those are the areas that are less well known. But the models, I mean, we can model into shallow waters, but the problem with shallower waters is they can become very complex. So the sort of shallower region is more complex in terms of tides and current speeds. It's much more variable. So these models work quite well in deeper water because it's less variable, right? But you can model into shallow waters. And the broad scale habitat maps, they do go into shallower waters. But my expertise is deep sea, and so my focus is back in the deepest areas.
Okay, thank you.
You touched earlier on the fact that you also have to include the pelagic environment. But most of this mapping is done on benthos, correct? How do you, especially when you're extrapolating typical benthic areas, how do you actually incorporate pelagic as well?
So we normally would map the benthic and pelagic separately, and that's because the things that drive organism distribution, as you know, Bronwyn, sorry, are different in the benthic and the pelagic. So in the pelagic, you know, the important things are things like frontal systems or upwelling areas, and that's really quite different to the drivers on the seabed, which are often substrate type and
yeah, I'm thinking more of the sort of benthic pelagic coupling between species.
Yeah,
and not so much the physical chemical stuff, but the actual connection between benthic and pelagic species, which of course is critically important for the fisheries.
Yeah, absolutely. So I think no one's quite cracked this yet, but I think what we are moving towards is spatial maps for the benthic, spatial maps for the pelagic, but then temporal maps that show that benthic-pelagic coupling, because that's a more temporal consideration, I suppose, and how things evolve over time. But what you have is temporal models for each habitat type, okay, that are able to show those relationships and that three-dimensional structure. But I will confess, I mean, we've not cracked it, and I'm not aware of anyone who has worked out how you bring these things together in a sort of spatial management framework. Well, it's not just spatial management, it's management framework. because you've got both spatial and temporal and the linkages to deal with, and it becomes very complex. So actually, interestingly, under Mission Atlantic, the EU-funded project that I'm part of, that's what we're trying to do at the moment, is link the spatial with the temporal, which includes that benthopelagic link, particularly in terms of nutrient cycling anyway, and to see if that's going to be possible. But yeah, it's complicated, but and I don't know if it's going to work.
Kurt has another question. Go ahead.
Yes, I was wondering about the scale.
Yeah.
You say you can predict ecosystems, but up down to a certain scale that you can observe. Right? Couldn't you use remotely operated vehicles to just see what's there? Do you get more information from trawling?
No, certainly for the vulnerable marine ecosystem work, the information we have on presence of some of those, particularly the cold water coral systems, all comes from remotely operated vehicle or towed camera systems, because they're the only real way to confirm you've definitely got a vulnerable marine ecosystem, because you can see it, as opposed to the trawl information, which tends to give you those indicator species, but doesn't necessarily tell you if you're in a vulnerable marine ecosystem. So certainly in terms of mapping, the new technologies of particularly autonomous underwater vehicles, being able to move around and collect imagery on the seabed will really help us to start validating these models. But I think we're quite a long way away from that. But yeah, definitely imagery is really, really important as definitely as a less destructive method, because you don't really want to be trawling your vulnerable marine eco.
No, thank you, Kerry.
Does anyone have any other questions? But I mean, Carrie is available by email, so you can always, I'm sure she won't mind. Maybe you can find her data, that would be great.